Welcome to the first episode of our long COVID series. Today, we are diving into the fascinating and complex pathophysiology of long COVID. What's actually happening inside the body of someone with long COVID? Why do some people recover fully, whilst others are left battling symptoms for months or even years? Let's break it down today. So what is long COVID? Let's start with a quick overview. Long COVID affects an estimated 10 to 30% of people who recover from acute COVID-19. Symptoms range from fatigue and brain fog to respiratory issues and cardiovascular complications. But what causes these? Today, we'll explore the leading theories and research breakthroughs explaining these lingering effects. Our first stop, persistent viral reservoirs. Emerging research suggests that SARS-CoV-2 does not completely leave the body in some individuals. Instead, the virus can hide in immune-privileged sites, such as the gut, lymph nodes, or even in the central nervous system. A study published in Cell Reports Medicine in 2024 detected viral RNA and proteins in intestinal biopsies taken months after infection. These viral remnants may provoke chronic immune activation, acting like embers in a fire pit, causing a persistent inflammatory response. This mechanism explains symptoms such as fatigue, low-grade fevers, and generalized inflammation. So we're looking at systemic symptoms here, seen in long COVID. This would be the primary clinical takeaway. Our next theory, mitochondrial dysfunction. Now let's shift gears into the metabolism. Chronic inflammation can disrupt mitochondrial function, impairing adenosine triphosphate production and increasing ROS or reactive oxygen species. This mitochondrial dysfunction was highlighted in a cell metabolism study from 2024, which found mitochondrial DNA damage in long COVID patients. This explains profound fatigue, exercise intolerance, and muscle weakness, classical features of long COVID. Finally, let's address the gut dysbiosis. Gut dysbiosis, or imbalance in the gut microbiota, has emerged as a significant contributing factor to long COVID. A 2024 study found reduced levels of beneficial bacteria, such as Faecalibacterium prausnitzii, which are crucial for regulating inflammation. So what is the mechanism behind this? The dysbiosis can lead to a leaky gut, allowing bacterial endotoxins like lipopolysaccharides, LPS, to enter the bloodstream and fuel systemic inflammation. So what would be the clinical correlation? Patients with gastrointestinal symptoms such as diarrhea and abdominal discomfort often have significant microbiome changes linking gut health to systemic symptoms in long COVID.
Next, let's discuss immune dysregulation. One of the hallmarks of long COVID is the overactivation of both the innate and the adaptive immune system. Studies have actually shown that patients have persistently elevated cytokines such as interleukin-6, TNF-alpha, and interferon gamma, all markers of chronic inflammation. This has been highlighted in the Nature Immunology study that was published in 2024. On the adaptive side of things, researchers have identified autoantibodies targeting the body's own tissues, likely due to a mechanism that is actually also seen in other sorts of infections, namely molecular mimicry between these viral proteins and host antigens. Another theory is that there's T-cell exhaustion marked by PD-1 overexpression, which further reduces the immune system's ability to control inflammation. This dysregulation possibly explains symptoms like the joint pain that is present in some patients, or the small fiber neuropathies that some patients experience, as well as the systemic fatigue. You can think of this as the immune system running on overdrive, it trying to fight an enemy that's no longer there, but ultimately accidentally attacking the body itself. Next, let's also talk about the vascular complications. The SARS-CoV-2 virus binds to the ACE2 receptors that are present on endothelial cells and can lead to endothelial damage. This process can trigger the release of a von Willebrand factor and also tissue factor that can set off the coagulation cascade. A fascinating study published in Thrombosis Research in 2024 revealed that many of these long COVID patients have persistent fibrin amyloid microclots that resist fibrinolysis. These microclots can ultimately obstruct microcirculation and can cause hypoperfusion to some of these critical tissues within the body. The takeaway message from this part would be that this mechanism explains symptoms like the brain fog or the chronic fatigue, and as well as the exertional intolerance, which could possibly be due to reduced oxygen delivery to the brain as well as the muscles because of the presence of these microclots. Let's now move on and talk about neuroinflammation and the CNS dysfunction induced by long COVID. Long COVID frequently involves neurological symptoms like brain fog, as we have previously discussed, but also memory impairment and mood disturbances have been observed in some patients. These may be caused by cytokine-mediated neuroinflammation due to the elevated interleukin-6 and TNF-alpha that can cross the blood-brain barrier, activating the microglia and the astrocytes, leading to what's often referred to as the sickness behavior. In some rarer cases, SARS-CoV-2 RNA has been detected in the CSF of some of these patients, indicating a direct viral invasion of CNS tissue. This was also actually discussed in a 2024 article in Brain Behavior and Immunity.
Joining us today is Charles Ward, a fourth-year medical student and active PhD candidate who is conducting research on the mitochondrial dysfunction induced by long COVID. He will be discussing the key milestones that were achieved in understanding the pathophysiology of long COVID.
Thank you very much for having me, Rehana. So I think first of all it's important to really figure out where we started, and that was with understanding viral structure and biology. And thankfully, when the acute COVID-19 pandemic first hit, the virologists had been studying coronaviruses previously, so it gave us something to start with. But just in short, the virus is a positive sense single-stranded RNA virus with about 30 kilobases of genetic material. And what that really means is that once it gains access to the cell, the RNA is just directly translated without intermediate steps in a series of double membrane vesicles. And I think we've all heard about certain viral proteins now, like S or spike protein, which is the part of the virus that gains access to the cell through angiotensin converting enzyme 2 or ACE2 or TMPRSS2, which is a cell surface serine protease that's highly expressed in respiratory tract epithelium. But there's also other proteins like the E protein, which is the envelope, the membrane protein, and the nucleocapsid protein. And the long and the short of it is the virus gains access to cells in the respiratory epithelium, hijacks replication and translation, and then produces much more viral particles, which enables this continuing infection. But I think the interesting part was in terms of viral persistence, when pathologists doing autopsies on patients who had died or had long-term effects of viruses started finding viral material turning up in really weird places, often very long after the infection. It was both RNA and proteins, and it was too often just to be contamination. And generally speaking, genetic material isn't free within our organism. You know, it must be chaperoned at all times, because free genetic material is recognized as a damage-associated molecular pattern. It's an indication that something has gotten out from where it was meant to be, and it's recognized by pattern recognition receptors like toll-like receptors and nod-like receptors and RIG1 and all those molecules that we know about from immunology and pathophysiology. And the real question was, why is this material turning up 100 or 200 days or even longer after infection diagnosis, when normally this material would be cleared fairly quickly? And to try and get to the bottom of this, scientists turn to computational modelling, and the main technique that we're using is RNA GPS, which is a very powerful machine learning tool that's used to predict interactions between RNA and subcellular locations throughout the cells. So the first thing that scientists started to find, or predict at least, was that viral material was heavily interacting with mitochondria, which is fascinating because we start wondering what's going on there and why mitochondria. And I think traditionally we think of the mitochondrial compartment of the cell as these organelles that are conducting oxidative phosphorylation and the main kind of furnaces or energy drivers of the cell. And we used to think of them as these singular discrete organelles, but actually nowadays it's better to think of them as a sort of fluid continuum of fission-fusion events, this compartment of energy-dense material within the cell. And I mean, I actually think of them almost a bit like a lava lamp, the way they kind of morph and fuse, and that's controlled by a series of proteins which are very highly conserved in all eukaryotes, from yeasts through to mammals. And really it's a part of homeostasis. It's balancing energy production of the cell with the requirements of the tissue in response to intra and extracellular factors. And like all pathophysiology, once that balance becomes out of control, it's where we start to see pathological elements of disease take place. But again, something that we hadn't really taken into account was the amazing capacity of mitochondrial proteins to influence the innate immune system, that non-specific immune mechanism that's activated when viral material enters our cells. And actually this kind of makes a lot of sense, because just like loose viral material activates the innate immune system, so does mitochondrial DNA external to the mitochondria. That's also a damage-associated molecular pattern. And it sets off a pattern recognition cascade, one result of which is the production of interferons and pro-inflammatory interleukins that, through a JAK-STAT pathway, produces inflammation, macrophage polarization, and ultimately in severe acute COVID leads to a cytokine storm. But in the longer term, the innate immune system also includes defective organelle replacement and repair apparatus. And the reason for this is mitochondria are producing reactive oxygen species all the time, and these cause oxidative damage to proteins. And these malformed proteins deform mitochondria and other subcellular structures, causing depolarization of the membranes and eventually leak of matrix material. And that leak in the most severe sense is recognized as apoptosis. But in a less severe sense, it also means that mitochondrial DNA and other mitochondrial proteins get where they're not meant to be going, and that activates inflammatory processes subsequently. And this is all really controlled through the PINK1 Parkin pathway, which is a method of ubiquitilating defective mitochondria when their membrane is depolarized. And they effectively get enveloped in a big membranous vesicle called a phagophore, and that then fuses with a lysosome, which has all the enzymes so that it can be degraded, and then the contents can later be recycled. And this is the backdrop of viral interaction with mitochondria. So we started asking questions about how viruses interact with that highly conserved apparatus of mitochondrial recycling, flux, and fusion-fission events. And we found really that viral genetic material interferes with mitochondria at pretty much every level. Both the production of interferons, the ubiquitilation of defective mitochondria, and finally the fusion of the phagophore lysosome autophagophore complex. And this really led to even more questions, and every step that we went through, we kept finding more and more questions that needed to be answered. And ultimately we turned to immunohistochemistry, which allowed us to localize viral genetic material and proteins inside mitochondria. And furthermore, mitochondria surrounded by double membranous vesicles holding viral peptides and RNA, which were grouped around these defective organelles. So, I mean, the long and the short of it is that in addition to disruption of innate immune system processes and interferon production, viral material is able to hijack mitochondrial flux, which prevents this normal fusion-fission degradation of these defective organelles. And the result of that is a retention of defective mitochondria sequestering viral material, which is then safe from degradation by normal autophagic processes.
And what would you say is the significance of all of this?
Well, I think the fascinating result from this really is that the symptoms are correlating with tissues that have high oxidative phosphorylation requirements. So when we think of the symptoms of post-acute COVID or long COVID, we think of, brain fog and neuronal CNS symptoms, and actually that's sort of correlated with the fact that we find a lot of viral genetic material in CNS tissue during autopsies. And then cardiac and muscle pain, those tissues are notorious for their high mitochondrial load and high OXPHOS requirements. And then most troublesome of all, I think, is the generalized fatigue, which I know a lot of patients find it very hard to be taken seriously when they talk about generalized fatigue, but actually this is a symptom that's emblematic of disrupted energy homeostasis throughout all tissues of the body. And I appreciate this seems very academic at the moment, but actually it's one of the great things about pathophysiology is that really this is the map that we use to navigate the treatment terrain, because it allows us to open up new fields of treatments and new modalities or new pathways that can be modulated, and that really is what this is all about, I think.
And finally, I'd like to ask you how you got involved in this type of research on mitochondrial dysfunction induced by long COVID.
Well, I'm very fortunate in that my supervisor, Professor Beate Schlichtholz at the biochemistry department, and I have been able to study some serum samples from patients with long COVID, and that includes patients with fibrosing diseases or previously had idiopathic pulmonary fibrosis, and also just control subjects. So we've been assisted by a generous grant that allows us to interrogate the metabolomics, the sort of metabolic picture of what's happening on a biochemical level with those patients. But really it's led us into some much more fruitful work that I think we've all been very surprised about, because behind the scenes we start to see that once we interrogate this concept of mitochondrial flux as the primary homeostatic means of balancing the metabolic profile of the organism, actually the disruption of mitochondrial flux is a background and behind the scenes picture in a wide and diverse number of disease states, including things like Parkinson, asthma, especially with airway remodeling, viral hepatitis, fibrosing diseases of all sorts such as idiopathic pulmonary fibrosis and hepatic fibrosis and systemic sclerosis. And it's almost like we're just starting to scratch the surface of this whole new lens through which we can view disease processes within the body. And I think really what it's going to lead us on to is some really good and really fruitful large-scale metabolomics studies where we can get into the nuts and bolts and see which metabolites are altered at various stages and start to see patterns or contrasts with certain disease processes and really understand how the metabolic profile of the organism is altered during disease states, and that hopefully will lead to firstly a much greater understanding of the pathophysiology of a wide variety of diseases, but also enable some really quite interesting advances in treatments and therapeutics throughout that work. So I think really the future is quite bright for us.
Thank you so much. There definitely seems to be a gap in research regarding this area, and it's extraordinary that you guys and your team in the biochemistry department are trying to bridge this gap. Thank you for the insightful and comprehensive explanation, and we were really glad to have you here today.
No, thank you for having me.
Alright, so joining us next is Alexandra Oppmann. You guys already know her as Ola. She's the head of the Public Health Student Scientific Circle, a public health graduate, an active PhD candidate, and a fourth-year medical student. We are thrilled to have her here to allow us to see things through the lens of public health.
Thank you for letting me join you guys today.
So Alexandra, I'd like to ask you about the prevalence and identification of long COVID. How do you think long COVID manifests in different populations, and what possible challenges exist in identifying and diagnosing it consistently across diverse demographics?
So long COVID presents with a wide range of symptoms, including fatigue, cognitive dysfunction, respiratory issues, and it varies across populations. In a 2023 Nature review, in fact, more than 200 symptoms were identified, impacting multiple organ systems, and at least 65 million individuals globally are estimated to have long COVID. So the challenges in consistent identification and diagnosis stem from the lack of standardized diagnostic criteria. Obviously, it's hard to standardize criteria when you have around 200 symptoms. The variability in this presentation of these symptoms in people, and disparities in healthcare access among different demographics. In 2024, in October, the British medical journal Global Health conducted an observational study across 17 countries, and generally speaking, they found that long COVID does affect populations globally equally. Across these 17 countries, they had tagged them as high-income countries and low-income and middle-income countries, but they also emphasized that it was difficult to access the data on low-income and middle-income countries in comparison to high-income countries. So that continues to show the need for global characterization of long-term sequelae to address these challenges.
So that's quite interesting. You talked about the socioeconomic status of this type of situation. What do you think the primary risk factors associated with developing long COVID are, and how do these social determinants of health, such as socioeconomic status, race, and access to care, contribute to these primary risk factors?
Yeah, so primary risk factors for developing long COVID include severe initial infection, being older in age, and having pre-existing health conditions like autoimmune or chronic health conditions. Social determinants like lower socioeconomic status and racial disparities, as well as limited access to healthcare, will exacerbate these risks by influencing the exposure rate, healthcare quality, and the ability to manage these chronic symptoms. Research highlights that there's an importance of addressing the social determinants to mitigate long COVID's impact on patients.
And are there disparities in how long COVID is diagnosed or managed among different populations and different communities? If so, what steps can public health systems take to address these disparities and inequities?
As always in public health, disparities will exist in both diagnosing and managing long COVID, especially affecting marginalized communities. Factors including limited healthcare access, like I mentioned before, the socioeconomic challenges, and there's also potential biases within healthcare systems. In the Nature review paper that I mentioned before, for example, they found that there was a higher prevalence of long COVID reported in certain ethnicities, specifically Hispanic or Latino heritage, and the socioeconomic risk factors included lower income and the inability to adequately rest in the early weeks after developing COVID-19, which is something that most people don't think about on the day-to-day, I think, but even for a family of maybe 5 with a single parent who's working 2 jobs, somebody like that is already at a lower, it's already at a challenge to find the time to even rest when they're sick with COVID, let alone developing long COVID symptoms. Public health systems can address these inequities by increasing awareness and improving access to specialized healthcare, as well as ensuring culturally competent healthcare services. In 2024 August, there was a British Medical Council public health study published which examined long COVID impact on Black and Latinx communities. They concluded that there is a need for multilingual, accessible information about long COVID symptoms and improved outreach and healthcare delivery, which essentially underscores what we've been talking about, the need for equitable healthcare approaches.
So we definitely have a long road ahead of us, but let's also talk about preventative measures. How can we just prevent this from happening in the first place? How effective are public health interventions like vaccination campaigns in preventing long COVID from happening in the first place? And what additional measures might help to reduce its prevalence?
So even though maybe we could call it the emergency phase of COVID is over, but the virus continues to spread globally and is still endangering people's lives, especially, like I said, people who are older or who have chronic diseases, who are immunocompromised or pregnant. The World Health Organization has established as of October this year that the vaccines are safe and effective. And in 2021 alone, the vaccines saved at least an estimated 14.4 million lives worldwide. So we know that vaccination campaigns are effective in reducing the incidence of long COVID by preventing severe acute infections. Additional measures include promoting booster vaccinations, encouraging public adherence to health guidelines, and implementing early treatment protocols to mitigate the long-term effects. I think something that everyone can do, whether they are a healthcare practitioner or a public health service member, is also just reporting misinformation on social media. This is a really easy way that every average citizen can get involved in when they see something online that's not correct, especially about vaccinating or masks. They can use their own accounts to report it. So that's an easy way for people to get involved in preventive measures.
Alright, and finally, let's discuss policy and community support. What policies or community-based initiatives could public health leaders implement to support long COVID patients in their recovery and their reintegration into daily life? And what other possible policies could they introduce in order to achieve herd immunity at a much faster pace?
So policies to support long COVID patients could include things like providing access to multidisciplinary rehab centers, implementing workplace accommodations or like paid leave, ensuring social support systems are in place. I think this is probably the most important part. People heal better when they feel supported by their community. Community-based initiatives could be things like support groups or educational workshops and community outreach programs to increase awareness and offer tailored resources for patients' needs. All these initiatives could also include collaborations with local organizations like public health offices or like local clinical offices to maintain mental health support, peer mentoring programs, and accessible physical therapy to aid in recovery. By fostering a supportive community environment, public health leaders can help long COVID patients reintegrate into daily life while addressing their physical, emotional, and social challenges.
Thank you so much for that thorough explanation, and we were very happy to have you here today.
Thank you so much.
We hope that you thoroughly enjoyed it, and more importantly, that you learned from it.